auspicious moment to start.
Okay. So, on behalf of the Department of Material Science and Technology, the department which belongs under the School of Natural, Applied and Social Sciences, Macau, West Bengal, I, Dr. Anirban Roy, is welcoming to each and every one of you for being here with us today. So, our today's session is on materials characterization, and in this session, our expert speaker will speak on absorption, diffraction, and other useful techniques used for materials characterization. And I had to share on another thing that previously we had arranged.
Just an announcement. If you are not speaking, please keep yourself muted. If you are not talking, please be cooperative, keep yourself muted. Thank you.
Yes, please. So previously we had arranged two similar type of sessions where one expert speaker had spoken on electron microscope, and in another different session, another speaker had shared her views on UV visible spectroscopy. So hopefully after this session, the participants or the students completed their knowledge required for materials characterization. So this is the main motive behind this session. And at the very beginning, I would like to have the privilege to introduce our today's expert. So today we have with us Professor Dr. Sukhendu Samajdar, Director of the School of Natural, Applied and Social Sciences. He is also the head of the Department of Material Science and Technology. Then we have, I don't know if she has joined or not, Ms. Monalitha Chat. She is not joined yet. Hopefully she will join. She is the assistant professor of our department. And as an expert speaker, we have with us Dr. Rudra N. Samajdar from National Physical Laboratory, UK. So at the very beginning, I would like to request our HOD sir to say a few words and introduce our today's speaker and let him start our today's session. So over to you, sir.
Thank you, Dr. Roy. Namaskar. Welcome on behalf of Maulana Abul Kalam Azad University of Technology. It's my privilege to welcome one and all of you who are attending today's session. I am actually quite pleased to see the size of attendance that speaks a lot about your interest in the subject, as well as our effort to promote learning in this unusual condition. So once again, my respectful welcome to the borderless learning community. Before I get to introducing today's speaker, let me make a little bit of a housekeeping announcement. This session, this series, the materials characterization series, is actually organized as a part of a guided learning program, especially relevant to the second year, to a second year course in the second year of the MTech in Material Science and Technology that we offer here and running successfully for the last few years at this university, Macau. So for that is the primary center of this purpose of this. However, we typically open this up to the general audience, which we have done today as well. Now, especially for those students in the second year of the MTech, who are taking the course, please take a note that this is very important that you attend this carefully and take special notes and incorporate your learnings from today's session into the reports that you have prepared and are preparing. Now, moving over to the introduction of today's speaker. Dr. Rudranarayan Samajdar is a blooming young chemist working at the National Physical Laboratory, United Kingdom, for nearly 2 years on the material science applied to modern battery and energy systems. However, while introducing Rudra, I must not miss two aspects. As a young scientist, once again, please keep yourself muted if you are not speaking. As a young scientist, he is exceptionally well read in the fields of humanities, and perhaps as a corollary, but equally. Please keep yourself muted if you are not speaking, please. I request all of you to cooperate. We have an egalitarian platform with no special privileges. So please respect that and keep yourself muted if you are not speaking. And because of some of these disturbances, I have to start over the introduction once again. Dr. Rudranarayan Samajdar is a blooming young chemist working at the National Physical Laboratory, United Kingdom, for nearly 2 years on the material science applied to modern battery and energy systems. However, while introducing Rudra, I must not miss two aspects. As a young scientist, he is exceptionally well read in the fields of humanities. And perhaps as a corollary, but equally valuable in its rarity, is that Rudra's communications and presentations are typically garnished with touches of philosophy and humor. Hopefully, today's will be no exception. Welcome to the session, Dr. Rudranarayan Samajdar. The podium is all yours.
Thank you, Professor Samajdar. Thank you, Dr. Roy. It's an honor and privilege to be here with you all today. I'll quickly try to share my screen to get this going. Right. I guess you're able to see my screen, right?
Yes, it's visible.
Yes, visible.
Perfect.
Yes, full screen presentation mode.
Just a second. Right. So, yeah. Morning, everyone. The title of my talk today is Lighting Up Matter Using Absorption, Emission, Diffraction, and Other Useful Curiosities. My name is Rudra. I'm working at the Electrochemical and Electromagnetic Technologies Department at the National Physical Laboratory at the United Kingdom. So I'll be talking about absorption, emission, diffraction, a couple of other sort of related techniques which are quite useful to most practicing material scientists and spectroscopists around the world. Parts of it may overlap with things that you've already read, and feel free to stop me if you have any questions, because when I'm on the presentation mode, I'm not able to see if you're raising your hand. So just feel free to unmute yourself and ask a question if there's anything that you'd like to raise while I'm giving the presentation. I'll quickly start the talk. Atoms, molecules, materials, they essentially have sort of secret lives of their own. So, as you know, Feynman has this very famous quote, which says that everything that living beings do can be understood in terms of the jiggling and wiggling of atoms. And this is from lectures on physics, a 3-volume sort of magnum opus on physics that many of you might have come across or started reading. I like to connect this with very famous dialogue from the movie The Lion King, where the original Lion King, Mufasa, tells his son Simba in this very sort of evocative scene in the movie that everything the light touches is our kingdom. You know, to be honest, I never sort of related this statement, everything the light touching is our kingdom, to spectroscopy in general. But I had this discussion with a colleague of mine, one of my batchmates from ISC, who's now doing a postdoc at the University of California at Los Angeles. And she pointed out that this sort of is valid for all people who practice spectroscopy, because effectively what you do is that you try to look at things with light, and that may be visible light or other forms of electromagnetic radiation, but light after all. So as practicing spectroscopists, we try to sort of bridge a domain, build a bridge here, using light to understand jiggling and wiggling of atoms to make sense of the world around us. So the sort of approach that I guess everyone wants to take when using spectroscopy to understand material science is to first of all understand atoms, molecules, materials at a very sort of basic fundamental level, and then see it using spectroscopic techniques. And then once you're able to see something, you actually measure, and that's understand what you're seeing in terms of fixed observables. And once you're able to measure, you're able to sort of deliver products based on these measurements and things that you're seeing. So you see that there are three things that's there in the slide. So I'm guessing most of you will be able to recognize this is a carbon nanotube. For those of you who are working with nanomaterials, and especially carbon nanotubes have emerged as a sort of hot topic in nanomaterial research, having a wide variety of applications. This is a carbon nanotube as I drew from ChemDraw. There are various types of this molecule. This is one particular type. The picture on top is quite sort of a picture that's quite pertinent with the current times. This is essentially the receptor of the spike COVID protein. That's the spike protein of the coronavirus that is binding to the angiotensin converting enzyme, that is the ACE2 that's present in our that's present in our body. So essentially, this is the coronavirus binding to the protein in our body, and thus this is one of the sort of defining moments in which the virus starts infecting the body. This is measured using cryo-electron microscopy. And the thing that you see here is a chemical structure. It has lithium, which is purple in color, oxygen, which is red in color, and cobalt, which is blue in color. This is lithium cobalt oxide. And this is important because probably it is in most of the lithium-ion batteries in your cell phones that you're holding now. So this is the sort of atomic or molecular level or material level understanding of the world. And when you shine light on this and understand what you're seeing, you get deliverable products, such as, you know, if you know the structure of the coronavirus using cryo-electron microscopy, that helps you actually design vaccines, which have a tremendous impact on the well-being of the human race. If you know CNTs, then you can actually make materials with CNTs. You know, around about 10 years back or so, there's this Swiss bike company called BMC, who were making bicycles called the BMC Pro Machine, which were made with CNT, and they were known for their very lightweight and weight and high tensile strength. This one, the lithium cobalt oxide, is now commercially available as batteries, and this is the Sony BP U70, you know, which is a commercially available product. So spectroscopy or light matter interaction essentially helps you transition from this domain to this domain. Now, when we start talking about spectroscopy, the first thing that you would ideally want to know is how to represent molecules. Representing molecules means it's somewhat like the transition from geometry to analytic geometry, where you have a molecule, but you should represent it. You should be able to represent it using coordinates in order to understand molecular motions, the so-called jiggling, wiggling, better. So there are two ways of representing molecules, which I'm sure most of you have already come across at some point of time in your undergrad or postgrad studies. And the first thing is The first system is Cartesian coordinates and the second system is internal coordinates. So Cartesian coordinates, essentially you put a molecule, this is water, oxygen and 2 hydrogen atoms, and you put it in Cartesian space and assign XZ coordinates to all 3 atoms. When you go to internal coordinates, the sort of advantage of internal coordinates over Cartesian coordinates is you don't need to define XYZ, but you can define parameters which are much more closely related to the molecule in itself. And for example, in this molecule, this is water. So the internal coordinates are oxygen hydrogen bond lengths, the ROH in these 2 systems, and the bond angle. So effectively, you can represent molecules using XYZ coordinates, which are Cartesian, or bond angles, bond lengths, dihedral angles, which are internal coordinates. And once you know how to represent this, you need to sort of feed it onto a computer, because a computer will help you visualize molecular motion. So how do you feed it to a computer? There are 2 ways of doing this. Again, for those of you, I'm assuming for those of you who are doing computational chemistry courses, this is a fairly simple thing to do. So there's something known as a Z matrix, which you can design for a molecule. And this essentially consists of the bond at the atom centers, the bond lengths, and the bond angles. There's a fixed sort of way of putting this up. And if you can design it, you can just write it in Notepad. It's just look up how to set up a Z matrix, fairly easy. If you can't, if there's any sort of queries that you have, get in contact with me over email. But yeah, you can just write up a Z matrix for something as simple as water, knowing bond lengths and bond angles. And if you feed this to a standard computational chemistry program, most of them are available free, open source, freely downloadable from the internet. You can actually visualize the molecule in the program. Another way of doing this or representing molecules is using XYZ files. And that's essentially, you have 3 atoms, you put in the XYZ coordinates, and this is the XYZ file for water. So effectively, if you take a simple molecule like water and want to represent it in your computer, and instead of drawing it out in ChemDraw, these are the 2 standard ways of doing this. And why do we need to set up Z matrices or XYZ files? Because essentially you can draw a molecule in ChemDraw, or even you can sort of schematically represent it in PowerPoint. The reason for doing that is that, taking you back to, around about 2013, as you might have, you're probably aware, the Nobel Prize in Chemistry went to Karplus and co-workers on development of computational methods for understanding chemistry. So there's a very sort of deep statement that Martin Karplus made. You might be aware of Karplus's name from your course in organic stereochemistry. There are these Karplus angles when you do conformational analysis. However, what Karplus said was that the computer is your Virgil in the world of atoms. Virgil was essentially the poet who guided Dante to purgatory, if you've read Dante's poetry. So the computer is a very, very sort of crucial guide that you do need if you're going to understand spectroscopy. So that's why it's good to know how to do this. I'll just come back to these a little bit later when I talk a little bit more about molecular motions. So now that we know how to represent a simple molecule such as water, the next thing is to understand molecular motion. So again, this is a slide that probably most of you are aware of from your undergraduate classes. So if you have a 3 atomic system, like this is water, you essentially have 3 n degrees of freedom. That's the sort of general physical chemistry. So out of which 3 are translational degrees of freedom, as you see here. This is like X, Y, Z. So the molecule can translate along the 3 main axes. Then you have rotational degrees of freedom, which is basically the molecule rotating along the axis. So it can rotate along the y-axis, it can rotate along the x, it can rotate along the z. I've taken a right-handed coordinate system here. So you effectively, if you do x plus y, you end up with z. And then you have vibrational modes of motion, where basically the number of vibrational modes are 3n minus 6 if it is a nonlinear molecule, because effectively you can rotate along all 3 axes. However, if it's a linear molecule, then when you have something as sort of, it's like, if you can see the molecule that I'm holding up in front of the camera, it's carbon dioxide, right? So this has a sort of axis which passes along the center of the molecule. And if you rotate along this axis, it doesn't make a change. So effectively, then one molecular axis goes away, and you are ending up, you end up with 3n minus 5 degrees of rotation. So just sort of taking a cheeky pun at one of the common statements in the literature that man as mankind as well as molecule are condemned to be free. So we would want to understand what sort of freedom is given to molecules, so that, and using that, we try to understand spectroscopy in general. So going a little bit more into detail, so there are certain things that are known as normal modes. So this is effectively, a little bit more, looking a little bit more into detail on, the kind of vibrational spectroscopies that one would tend to do. So for example, for a molecule like water, which I've been talking about here, it has 3n minus 6. So that makes 9 minus 6. That's they have 3 normal modes, right? Now, what are the 3 normal modes? So if you take the molecule like water, which I'm holding up here, I hope you're able to see the molecule. So the 2 bond lengths, they can change, right? So this is length and this length. And you can have symmetric stretching of both bond lengths, that's both change by equal measure at the same time. You can have asymmetric stretching, where one bond increases or compresses at a slightly different rate compared to the other one. And then you can have a change in this angle. So these are the 3 normal modes associated with water. If it's carbon dioxide, then because it's 3n minus 5, so there are 4 modes associated, vibrational modes associated with it. There are symmetric modes, asymmetric modes, and then the molecule can bend in the plane of the slide, and it can bend away from the plane of the slide. When doing this analysis, which is known as a normal mode analysis, done routinely by most rotational vibrational spectroscopists all over the world, you tend to take advantage of the symmetry properties of the molecule. Now, for example, for this molecule, for those of you who have done a little bit of group theory and point groups, so there is a C2 axis here, which basically if you do a C2 rotation along this axis, you're going to end up with the same molecule. And there are 2 planes of symmetry. So this is why this is known as a C2V point group. In case this terminology is new to you, you will be learning about this soon in your spectroscopy course or as a part of your inorganic chemistry course. For carbon dioxide, the point group is known as DH. And this is because it has a sort of C infinity axis along this and a dihedral plane here. And if you know the point group of these systems, there are certain things known as character tables. Again, my apologies if you know about this already, but for those of you who don't, each point group is actually a collection of symmetry properties associated with the molecule. And the mathematical way of representing this symmetry properties is associated with something known as character tables. Again, spectroscopists use it routinely. And if you know this, then it's very easy to predict the sort of vibrational properties of molecules. So a quick sort of reminder here is that most of you will be like doing practical material science and spectroscopy. Mathematics is something that you essentially cannot avoid. So mathematical tools are indispensable in most areas of practical chemistry, even though we often tend to think that you can sort of deconvolute mathematics out of chemistry. In most places, this cannot be done. So having said this, I'll just quickly try to show you something. I'll go to stop share, and I'll try to show vibrational spectra of carbon dioxide, how you can sort of generate this in your computer. So just a second, I have to share another screen now. So, right. Are you able to see my screen? I hope you're able to. And do you see the carbon dioxide molecule here?
Yes, it's visible.
Yeah. So, I'd spoken about Z matrices, right? So this is a Z matrix representation of carbon dioxide. Extremely simple way to do this. Look it up over the internet and try doing this in Notepad. And then there's this software called JMOL. JMOL, it's essentially an open source software. You can download it from the internet. And once you have, now the next thing that you would want to do is essentially take this molecule carbon dioxide and do a vibrational mode analysis on the system. Now, the thing is that to do vibrational mode analysis, you do need specialized software. You can't do it using free software. But anyway, there's something known as Gaussian, and you need to feed this carbon dioxide molecule into the software, ask it to calculate vibrational modes, and then it just comes back to you with the data. But anyway, I've done this as an exercise and to show you all. And what I'll do here now is try to show how it vibrates. So essentially I click on start vibration. So probably you can see here that this is the first mode of vibration that's associated with the molecule. Now remember that there are 4 modes of vibration. So if I just again go to tools and click on the next frequency. So as you see, this is now bending in the plane of the molecule. So this is the second mode of vibration. Now, again, if I go to the next one, this is sort of bending away from the plane of the molecule. So this is the third mode. And here's another one. So as you see here, using a simple molecule like carbon dioxide and a little bit of access to computational chemistry software, a principal part of which is still open source, you can visualize these vibrations easily and try to understand your sort of physical chemistry / spectroscopy coursework. Right, so I need to stop this. I need to share my other screen, there's something called new share. And I come back to my presentation here. Right. As you see here, so this is what I was talking about, different kinds of vibrational modes, which are normal modes associated with molecule. Now, a quick question that you can ask here. Again, feel free to unmute yourself and ask a question during the talk, because while I'm presenting, I'm not able to see the chat box or the fact that if you're raising hands or not. An important sort of question that you might tend to ask here, that why are these modes called normal? So there's a little bit of sort of detail associated with this. I don't really want to go into this because this is quite a bit of technical vibrational spectroscopy, but just it would suffice you to know. I'm sure some of you know it already. If you don't, you can look it up on any standard vibrational spectroscopy text. But these are modes which are essentially independent of each other. So you try to identify independent modes of vibration, each of which has a specific amount of energy associated with it. Recall your undergraduate physical chemistry class where you had talked about the equipartition theorem, right? So normal modes are special kinds of modes. All vibrational modes may not be normal modes because these are sort of special independent modes which act independent of each other. So yeah, sorry for that sort of digression, but yeah. So these are normal modes of vibration, and so this completes, 3 major sources of molecular jiggling between translation, rotation, and vibration. So what are some of the other modes, which sort of motions which are associated with molecules? So then, for example, you have rotational spectroscopy, which I've already talked about. So when the molecule rotates, it sort of absorbs energy, and you can use spectroscopy again as you put energy in the molecule and see it rotate, and then use that information to find out specific things about the molecule. Rotational spectroscopy is an important branch of spectroscopy. I will not be talking about it today and tomorrow, for the main reason being, that it's a very special branch, and most importantly, you need to study it only in the gaseous phase, because once you're in condensed phases like liquids, then there are enough sort of intermolecular interactions in the sort of lattice which prevents sort of free rotation. And just suffice it to know, you might have read about this in undergrad chemistry from standard rotational spectroscopy texts like Bandel and McCash, maybe, or Hollis, that there are molecules are classified according to their moments of inertia into different kinds, like spherical tops, linear tops, and all of these have unique properties, and you can actually set up the the rigid rotator Hamiltonian and then solve for energy levels and thus understand this. However, just a sort of interesting point with respect to rotational spectroscopy is that rotational spectroscopy is essentially the basis behind microwave ovens, because rotational energy states are so spaced that microwave energy can help do a transfer from one state to another. So when you're heating your food up in a microwave, you're essentially taking the molecules to higher rotational energy levels. And because you're taking that, because of dissipation, you're having heat generated in the food, and that's the way. Apart from rotational spectroscopy, there's some other sort of modes of motion that are associated with molecules, such as, for example, diffusion. This is a physical phenomenon most of you know from your high school physics/chemistry, that when there's a concentration of molecules in one side and there's a sort of, very dilute kind of a regime on the other side, this is going to sort of move from one side to another. The physics of it is complicated. There's fixed laws, and you need to solve peculiar kinds of differential equations. But yeah, diffusion is a very common sort of phenomena that you see everywhere. This is something that molecules do. Another thing, for example, is fluctuation. For example, when you have a protein, for those of you who come from a biology/biophysics chemistry background, the proteins have these kind of long backbones, which are known as peptides. Like these have amino acids that I've tried to show here. So these are like amide linkages, right? So the CO and H2 linkage. And because of this is not exactly static as is shown here, right? So these molecules, the bonds have a certain degree of freedom associated with them, so they can fluctuate. So this kind of fluctuation can also be followed using spectroscopy techniques. I might be coming to them a bit later, maybe next day, tomorrow that is. And another thing is basically, when you take a molecule or you put a system in equilibrium, you add a perturbation and take it to a non-equilibrium state. And this can be something as simple as there are a set of dipoles in your system and you're applying an electric field and the dipole gets perturbed. And then once you take off the perturbation, it comes back. So this is known as relaxation. So relaxation is also another very important technique that's common in spectroscopy. And all of these phenomena, apart from your traditional, the 3 fundamental degrees of freedom, all of these can also be studied using spectroscopy techniques. I'll be telling you bits of these maybe tomorrow. And this tells you about molecules. Now, if you look more, if you look further into the more atomic level of things, then till now I've talked about bonds rotating, vibrating, moving, molecules moving from here to there. But what happens at a slightly deeper scale? So what happens is that you've got electronic energy states in atoms, and you can have something like electrons absorbing energy, showing absorbance, going to a higher excited state, coming down, showing fluorescence, or going to a different state and then coming down, which is known as phosphorescence. So if you look deeper into this, then there are these processes which occur inside the atom. And these occur always at ultra-fast time scales. The term ultra-fast became popular with, the introduction of laser-based spectroscopies, out of which you might have heard about Ahmed Zeeul, who was the pioneer of this kind of techniques. These help us actually understand bond making and bond breaking. And bond making, bond breaking, as you know, is sort of foundational sort of thing that governs chemistry. And for those of you who come from a chemistry background, you might have heard about transition state theory, where the transformation from one molecule to another goes through a sort of transition state in the energy landscape. And these kind of transition states involve bond making, bond breaking, and they can be observed by these kind of ultra-fast spectroscopy. And depending upon the type of energy that you're using, or the type of radiation, for example, you can use UV visible, you can use X-rays, you can use lasers, and many other kinds of stuff. It helps you probe different levels of energy like this. And if you do time-resolved experiments, that is, you're measuring the time associated with these kind of transitions, then you can also probe the time scales associated with it. So for example, you want to find out at what sort of space do bonds form in a molecule. They are in picosecond or femtoseconds and even faster. So if you can do time-resolved experiments, well-designed, they are difficult experiments, not simple ones, but you can actually find out information from them. So this is the sort of deeper picture when you go from the bond or the molecule level to a more sort of atomic particle level. Absorption by nuclei, by the way, is also possible. And this is the domain of NMR spectroscopy, which is fundamentally, present everywhere. I hope none of you have had to do this, but for example, if you ever had to go for an MRI for any kind of medical reason, NMR is the basis of MRI-based imaging in medicine. Following this, so I've talked about, vibrations, rotations, translations, more kind of macro scale phenomena like diffusion, fluctuation, and kind of more atomic phenomena like absorbance. So we come to this kind of a sort of broad picture. So at the very molecular level, you have something like this. So let's say bonds. This is again the sort of single asymmetric unit of lithium cobalt oxide. You have lithium, you have cobalt, you have oxygen. And then you have a bond like this. This is a peptide. At the next level, you have something like what is known as a unit cell in crystallography for those of you who are doing crystallography courses. And so this is lithium, cobalt, oxygen bound together in a systematic way, giving rise to a well-defined unit in crystallography. The equivalent in kind of the bio perspective is this kind of a peptide chain holding in a particular way, giving rise to what is known as a secondary structure in a protein. I've taken this structure from this particular paper, but for those of you who are aware of the way in which literature is represented. So this is the sort of journal name, this is the year, this is the volume, and this is the page number. My apologies if you know this already. But yeah, this is the sort of secondary structure that taken from this reference. This, by the way, is hemoglobin. And this particular crystal structure is taken, this is lithium cobalt oxide taken from this 1997 report on ionics. So this is the next level of things. So this is, you can think of kind of the atomic level, molecular level. This is the slightly sort of local molecule, a set of molecule or a stereochemistry arising from structure. And then when you go to the next level, this is essentially the sort of larger picture where you actually have a crystal packed. You know, if you take, you know, if somebody gives you a sort of a lump of sodium chloride in your hand, which, you know, you've compressed some salt in the kitchen and made it, that can have an arrangement like this. Obviously, it will not have lithium, cobalt, and oxygen, but it will have something which is macroscopic. And this, for example, is the hemoglobin protein. It is present in all of our bodies. We're using it, breathing in and out, binding with oxygen. And this is the sort of macroscopic scale. So the sort of question that you would want to ask as trying to, in one sense, enlighten matter is, how do you go from here to here? So what are the kind of techniques that you would want to use if you wanted to see something like this, and then later on something like this? Obviously, it's not going to be the same all throughout. It's going to change. But this is a transition that you would obviously probably want to do in a kind of small view to a large scale view. How to do this? Obviously, the answer is light, more light. So electromagnetic radiation is, you're all aware of this. So you can classify, this is a very nice sort of representation which I took from Wikicommons, and this is EM radiation. It is classified on the basis of its wavelength or on the basis of its frequency. As you know, energy scales directly as frequency. So as frequency increases, energy increases. As wavelength decreases, energy increases. So as you go from left to right here, there is increase in energy. And this is the sort of visible area of the spectrum: violet, indigo, blue, green, yellow, orange, red. So this is your VIBGYOR. Beyond red, you have infrared wavelengths like this. Beyond infrared, you have microwave, and beyond microwave, you have radio waves. If you go on the other side of violet, that is towards the lower wavelength but higher energy side, you have ultraviolet, you have X-rays, you have gamma rays, and so on. And this is the sort of entire set of things which are available at our disposal. So we use this. Molecules and materials at different length scales. This brings me to the first sort of pit stop, if you would like to use a racing analogy. So I've been talking almost continuously and there have not been any questions. So do you have any questions up to what I've said?
Dr. Shomas, there are a few questions are there in the chat box. So may I repeat it?
Yes, please, Dr. Roy, if that's okay. Thank you so much.
So Abhishek Poddar, he is our departmental student. He is asking, what is jiggling phenomenon?
Hi, Abhishek. Thanks for your question. Jiggling wiggling essentially is just a part of semantics. That's the way Feynman put it. But you can think of it that jiggling wiggling is essentially different vibrational modes associated with atoms. So it's not a technical term, obviously. It's just the way that Feynman had phrased this in his lecture. But essentially, jiggling and wiggling can be thought of as different vibrational modes associated with molecules. You might have heard about, these kind of more technical terms such as wagging, rocking, scissoring. These are typical vibrational modes associated with molecule, and jiggling wiggling is a sort of broad general term covering all of them in a more non-technical way.
He has asked another question. What is perturbation?
That's a good question. Perturbation is essentially, if you're from a sort of physical chemistry background, it's sort of, as the word literally means, it's sort of taking the system away from equilibrium. That's the general meaning of the word. However, from a more sort of quantum mechanical perspective, as you know, you have a system at equilibrium and you have a Hamiltonian associated with it, right? And then when you apply, let's say, an external disturbance, like for example, you have this molecule, which is water, and it has a Hamiltonian associated with it, and you can solve, and you can set up the Schrodinger equation and solve for its energies. Now, when you shine light on it, light is essentially electromagnetic radiation. It is a time varying phenomena, which in the simplest way you can represent as y is equal to y max sine of kx minus omega t, where k and omega are, the angular wave number and the angular frequency. So this, external if you apply to the molecule, takes it away from equilibrium. And this phenomena is known as perturbation. There is an entire branch of quantum mechanics which is known as perturbation theory, and only about it in your postgrad courses. And this actually helps us to understand spectroscopy in general. Thanks.
Thank you, Dr. Somasdar. No such questions are there. So if we get any, so we will notify you.
Sure. Thank you so much, Dr. Roy. So if all of you are happy, I'll just try to move on to the next set of slides. So to continue with this, again, when I'm talking, I'm not able to see the chat box, so feel free to raise a hand or stop me if you think that there's something you'd like to ask. Right, so we've seen the sort of different length scales of materials. We have seen different kinds of dynamic phenomena associated with atoms and molecules. And we now know that, we use electromagnetic radiation as a probe. So let's move on. Absorption and, a little bit of physics of absorption. So this particular diagram is very, very, very well known from all, quantum chemistry, spectroscopy courses. I've taken this again. There's a Creative Commons license available from Chemistry LibreText. It's also there. The same image is also there in Wikipedia. So, this particular kind of a potential, recall it from your physical chemistry course in undergrad or first semester postgrad. So this is known as a Morse potential, right? So this is the kind of energy state. Here you plot energy versus atomic or nuclear coordinate. So this is the variation of electronic energy as a function of nuclear coordinate. This one, let's say the E0 is the ground electronic state, and E1 is, let's say, the sort of first excited energy state. Let's assume for the timing that both of them belong to the same spin, that let's say they're singlet. If the spin changes, then the things become a bit more complicated, which probably we don't want to go into right now. And associated with the electronic energy state are these kind of closely spaced vibrational states. Note here that the vibrational spacing slightly decreases as we go from lower to higher energy. This is a sort of a feature of the way in which we understand vibration, and it arises from the mathematics of vibrational energy. So the thing that I'd like to draw your attention to is that, when we are shining light on a system, it is effectively absorbing energy. So absorption, as you know, was one of the sort of first phrases in the title of this talk. So absorption, emission, diffraction, a lot of other things that light and matter do. So when you are shining light, if the energy is, let's say, enough to drive a transition from one of the vibrational states in the ground electronic state of the molecule to one of the vibrational states in the excited electronic state of the molecule, then this results in, let's say, absorption, and you can see signatures in spectroscopy. What essentially happens is that there is a transition from the ground electronic state to a excited electronic state. vibrational states may change, and this kind of a transition is governed by something which is known as the Frank-Condon principle. I won't go into details of this because obviously this requires a bit of quantum mechanics and it's going to unnecessarily take up a lot of time. I'm not an expert on this, but suffice it to know that this kind of a transition is defined by what is known as a transition moment integral. And there should be a reasonable amount of overlap between the two vibrational wave functions for this transition to take place. I'll come back to this again a little bit later. Once the system has gone to higher energy, obviously it cannot stay there for a long time. So this is essentially what, coming back to your question, was one of the earlier questions was, what is perturbation? So this is essentially a kind of perturbation of life to the system. It's not in equilibrium anymore. Once it has reached there, it will try to come back again to its ground state because that's what all systems try to do. This is dissipation working in nature, so it comes back. So this is absorption and this is emission. And absorption emission, as shown here, is sort of like the fundamental basis of many types of spectroscopies that we use regularly. A little bit more on this. So I've spoken about the Frank-Condon principle. My apologies, this slide has quite a bit of text. So the way that one would want to do this is that you need to set up the Schrodinger equation for a stationary system, which is the system at equilibrium. You know that electromagnetic radiation is a traveling wave. So you set up the Hamiltonian in that way. For those of you who are aware of this, the Hamiltonian is the operator that you apply on the atomic sort of wave function to find out energy. For those of you who are not aware of this, my apologies, but do go back and read up a little bit on this. If there's anything that you would like to know, do send me an email and I'd be happy to help you. So the electromagnetic radiation is a traveling wave. You apply this electromagnetic radiation equation on the Hamiltonian of the system, and this is known as a perturbation. And it is time dependent because, as you know, electromagnetic waves vary in time, right? So that's sort of the general physics of waves. And so when you do this, what you end up with is known as time dependent perturbation theory. And the entire mathematics behind this is sort of the bedrock underpinning spectroscopy in general. And if you go moving a little bit more into time-dependent perturbation, I've not shown any equations because obviously it becomes boring and it doesn't make sense unless all of you would want to know this. So a little bit more into this, that if you do this kind of a time-dependent perturbation theory analysis, that is, you set up a Hamiltonian of the electromagnetic radiation, apply it on the Hamiltonian of the stationary state, solve for the energies. Remember, this is reasonably easy. It's not terribly difficult. You don't need things like tensors and density matrices and so on. It's worked out in Levin's text. For those of you who follow quantum chemistry by Levin, it's a fairly reasonable derivation, I think, in the chapter on time-dependent perturbation theory. It's there in many other textbooks as well, including, I've looked at Walter Struve, which also has a good discussion on this. However, a sort of detailed analysis will show you that there is something known as absorption, which is the transition from this state to a higher state. There is something known as emission, and there is also something known as spontaneous emission. So these are the three major things that can happen that come out of the system if you do a time-dependent perturbation theory analysis. There are coefficients associated with these phenomena, and these are known as Einstein's A and B coefficients. If you've done this course, my apologies, you know this already. And just a small sort of comment on this is that absorption and simulated emission, sorry, this is a typing error. It's not simulated, but stimulated. So absorption and stimulated emission are basically what we see everywhere in absorption emission spectroscopy. Spontaneous emission is something a little bit more complicated. The physics is more complicated, and this is associated with lasers. So for those of you who've heard about lasers, laser spectroscopy is sort of based on this. So if you do this, you come up with these kind of coefficients of absorption, which are sort of related with transition probabilities or oscillator strength. And you'll find out the meaning of what is known as transition dipole moment or transition moment integrals. So what are transition dipole moments or transition moment integrals? They're sort of a measure of the probability of transition. And you'll find out that a measure of this probability of transition is something very similar to what are known as absorption coefficients in spectroscopy, in at least practical spectroscopy. So this is why I've put a word that Lambert-Beer law is not as empirical as it sounds. For example, you've always read about Lambert-Beer as a sort of empirical law where the absorption is is epsilon CL, right? I'll be coming to this a little bit later. But yeah, this has a sort of theoretical, I mean, this was developed as an empirical law indeed, but there is a theoretical basis with which you can use to understand this law. And more importantly, in this analysis, you'll also come across selection rules of transition, where you'll get to know that there are certain things which are forbidden in the atomic world. And why are they forbidden? Then they're just a sort of artifact of the quantum mechanical rules which govern the world of atoms and molecules. And they are the sort of selection rules. And obviously, as with any set of rules, the sort of there are certain things which break the rules, and breaking these rules give rise to new forms of spectroscopy. Well, just as a follow-up to this, here's this nice kind of a description of the Lambert-Beer law from this particular, reference. It's an open source, it's an open access, paper in ChemPhysChem 2020. I'm happy to share the slide, and if you're interested, you can look up this paper. It's quite lucidly, they explain quite lucidly the sort of development of the Lambert-Beer law over time. So attenuation, that's basically you have radiation passing through a system, and there is, attenuation happening. That's radiation is being absorbed, and the radiation that comes out is attenuated. This has actually been around for a very long time. As you see in these time scales, in spectroscopy, it was Beer who developed it in around about 1852. In photochemistry, in the domain of photochemistry, it was around 1857. This kind of a similar law came up. And electromagnetic theory, which was the more sort of like you can think about the kind of ab initio kind of modeling or understanding of this phenomena. This started being developed around 1865. So parallelly, these kind of branches have developed. This is very common in chemistry. For example, things develop parallelly. And now, for example, beyond a certain period of time, we know that these have developed parallelly and we can try to reconcile them. Very common also between chemistry, at least certain parts of electrochemistry, for example, the work that I do, and chemical engineering. So you can see here that these laws have developed parallelly, and this kind of a TDPT analysis will let you know that they can all be reconciled into a single kind of theoretical framework. So enough about absorptions and emission. A little bit more if we go, then there's this another kind of emission. So this is, the phosphorescence that I was talking about. So if you go back here, the kind of image showed that there is absorption, and then there's a sort of, this molecule comes down, there's emission. Note here, something that sort of was in the fine print was that these two are in the same spin states. So for those of you who, my apologies for those of you who are not aware of spin, I'm sure most of you are. So spin is again a sort of, it's not literally the sort of like something spinning, but it's a sort of quantum mechanical property associated with the electron. And as you know, from the Schrodinger equation, you can find out the principal, the azimuthal, and the magnetic quantum numbers. But the spin quantum number is something quite different. And understanding this requires a sort of detailed analysis of Dirac's model of the electron. I won't go into this, to be honest. I'm not an expert. I don't know much about it. But suffice it to say that there are two different spin states in most, it can be a singlet state and it can be a triplet state. So this is a kind of Frank-Condon principle diagram. The diagram is a bit hazy. I've taken this though. This is a very interesting, wonderful book on photochemistry by Krishna Kamini Rohatgi Mukherjee, who was, I think, a faculty at Jalapur University. It's one of the best books on photochemistry written by a Bengali author or by an Indian author. But anyway, this is I've taken the liberty of copying this image from here. And what happens is that there is an absorption to a sort of excited state which has the same spin as the ground state, and then there is a crossover to a different spin state. Now remember that this is a forbidden phenomenon. Quantum mechanics does not allow this to happen. But under certain cases, the singlet to triplet transition can be seen at low temperatures and condensed phases where the matrix is rigid and the temperature is low. Then it can go to a triplet state, and from the triplet state, it can then radiate energy. And obviously, as you can understand, the wavelength is going to be quite different from the emission that would have happened had it taken place from the excited, singlet state. And this phenomenon is interesting. There are many chemists who actually study that. This is known as phosphorescence, and this has a sort of physics spectroscopy associated with this, which is sort of independent. But I won't go into this except for the fact that I'm pointing it out. I just wanted to point out this phosphorescence is also another another form of emission spectroscopy, the other emission, if you will, that can be seen from molecules. Just a quick comment here for those of you who are chemists from a chemistry background, that this kind of an overlap between electronic energy states, kind of a singlet to triplet state, as I've said, occurs only at low temperatures and in condensed phases such as liquids or solids. Solid state phosphorescence is pretty common. You see lots of papers in the current literature associated with it. So this kind of a framework is also quite similar conceptually. Pardon me if this sounds a bit too far-fetched, but this is associated with Marcus's theory of electron transfer. For those of you who are doing special papers in inorganic chemistry and are dealing with electron transfer theories such as outer sphere, inner sphere mechanisms, there's this sort of foundational theory on electron transfer put forward by Rudy Marcus, who's a Nobel laureate, is currently based at Caltech, I think. And this Marcus theory also involves this kind of a transition between two states, and these are energies. The energies associated with them are known as the free energies of electron transfer. But yeah, the framework is sort of similar. And why I wanted to say this was that there are many models in chemistry. The good thing is that you can sort of draw parallels in trying to understand different systems. So yeah, this is the other emission. And now I'd like to come to something more sort of practical. So this is again, apologies, this slide has a lot of text. Sorry for that. And I didn't want to put in too many animations because of, sorry, my alarm is ringing on my phone, sorry. I didn't want to put too many animations in order to make it. So, UV visible spectroscopy is something that you've all done. I'm sure part of your assignment and as a part of your self-learning program. So recall the source and detector, you know the UV source, you know what a UV detector is, the geometry of data collection. So effectively, if you have actually done a UV visible instrument, a measurement, it's something like this. This energy going into, so you have a cuvette which is filled with a nice colored liquid, color samples show nice UV. So let's say you've taken a dye, any standard organic dye, maybe bromocresol or something. You dissolve it in water, you take it in a cuvette, you pass incident radiation through it. When the radiation that comes out is I0 e to the power minus epsilon Cl, where L is the length of this cuvette, C is the concentration of your dye, and epsilon is the absorption coefficient of the dye. So this is attenuation occurring inside. So this is your typical regular UV experiment. I'm sure most of you have done this or will be doing this, or at least have studied about it. So this is UV visible spectroscopy. I just wanted to point out, for those of you who have already done a kind of self-learning on this, that there's this kind of a physical model behind understanding UV. There's the particle in a two-dimensional box. If you solve for this, if you've done in your quantum chemistry course, then you can actually find out these quantum states. And these effectively can be used to model UV visible spectra of these kind of, these are known as annulines. So these are sort of cyclic molecules having alternative double and single bonds. And you can actually use a particle in a 2D box and use it to predict the UV visible spectra of two-dimensional molecules. However, keep in mind that there's something unique known as frost circle. I just wanted to say this. I couldn't help myself because you might have heard about this in your physical organic chemistry course. And what happens is that in a particle in a two-dimensional box, the bond length, for example, here I've shown you cyclobutadiene, right? So the bond lengths are assumed to be same. If they are different, then effectively there is a degeneracy, and these energy states, they become slightly different. However, not exactly related to this, but this is the sort of physical model which you can use to understand UV visible spectroscopy. There's just one thing that I would like to point out as a sort of follow-up through this, is that if you have powdered samples and not liquids, powdered UV is pretty simple, but if you don't have, liquid UV is pretty simple, but if you have powdered samples, then the physics is slightly different. There's no kind of transmission kind of experiment. This is what is known as a diffuse reflectance collection. So this is a sort of schematic that I've taken from Shimadzu's website. Shimadzu is a very well-known, UV visible spectrometer manufacturer. So the light goes in and there is a standard reflector, and then there is your sample. The sample tends to reflect the light, and this kind of reflection is known as diffuse reflection. It's one of the quirks associated with reflection physics, but diffuse reflection is slightly different from specular reflection. And you collect this kind of diffusely reflected light, and then you do a transformation which is known as Kubelka-Mook transformation. Sorry for this kind of jargon, but you might want to read about this in case you're doing a sort of project work or something on UV. And using this, you can then find out the absorbance, right? So there are two ways of doing this. That's what I essentially wanted to point out. The liquid way is fairly easy. The solid way is a tad bit more difficult. However, what is this used for? Again, this is used for understanding conjugated electronic structure, especially for those of you who would like to do work on nanomaterials. They're used to find out band gaps, which is the difference between conduction and valence band on molecules. And keep in mind that these occur on molecular length scales, right? Following absorption, the next sort of logical thing is emission, right? So emission spectroscopy is known as fluorescence spectroscopy. You excite at a particular wavelength the molecule and study the emission characteristics. And this is the kind of typical fluorescence spectra where this is essentially your absorption. It occurs, as you see, at lower wavelength, that is higher energy. And then once it is absorbed, then the molecule can emit a radiation at a slightly lower energy, that is higher wavelength, and this is known as an emission spectra. So effectively, there can be an overlap between an absorption and emission spectra. There may not be. So as far as spectroscopy is concerned, the instrumentation is similar to that used in absorption spectroscopy. The only difference is that you need to collect data at right angles, because if you collect at 180 degrees, then you'll be effectively looking at absorption. So the emission detector is usually kept here at right angles to collect the signals. And another thing that's important here is that by looking at the sort of decay of emission as a function of time, this is known as time resolved fluorescence, or the technical term is time correlated single photon counting. You can actually find out temporal information about chemical processes. So this is more in the domain of bond making, bond breaking that I was talking about. It's not ultra fast, obviously, but it can provide temporal information about chemical processes. So this is a sort of typical fluorescence decay. You take a particular wavelength and study the intensity of emission, and this decay is a function of time. I've taken this schematic from Bernard Valor's text, which is one of the sort of foundational texts on molecular fluorescence. And you can study this decay. You can set it up to a kind of an attenuation physics here. So e to the power minus t by tau. And looking at these kind of time scales, you can find out information about about what's going on inside your system. So information here, electronic structure, chemical perturbation, and this is something that you must keep in mind, is that UV visible absorption occurs from a ground state, so it is less susceptible to changes in chemical environment. Because fluorescence appears from an excited state, it's more susceptible because the molecules have dipoles and all this kind of weird stuff at an excited state, right? So it is more susceptible to changes in the chemical environment, and you can also find out molecular timescales from this. Having mentioned absorption and emission, I'll be talking about a case study. There's a small bit of work that I did in my PhD. So what I was trying to do was I took this kind of a molecule, which is known as a heme. It's essentially a kind of iron porphyrin molecule. And I was trying to look at interaction of these kind of simple amino acids with this system. Now, these amino acids coordinating with a kind of a heme. Heme is basically this molecule. It's known as hemine, essentially, for those of you who are aware. Coordinating to these kind of amino acids, when they coordinate to this kind of a moiety, this is essentially the sort of foundational structure of many protein structures. Now, for example, this is the UV visible spectra of the protein hemoglobin. So, this is the thing, plot absorbance versus wavelength, and there are signatures. And these signatures, as I pointed out earlier, are associated with the electronic conjugated structure of the molecule. So this thing is known as a Sore band. These are known as alpha and beta bands. And every molecule has their own sort of eccentric signatures, but they can be used to identify the molecule. So hemoglobin, which is a protein, and hemine, which is just a bare kind of an active site structure, as you see, have very different UV visible spectra. Why? Because their electronic structures differ. Hemoglobin has something coordinated here, and hemine does not have this. So this is one of the sort of an example of practically using UV visible spectroscopy to distinguish between the molecules. Now, for example, why emission becomes important is that, for example, when you're actually studying a dynamic process like the tryptophan molecule going and binding to this system, UV visible spectroscopy, because it appears from a ground state, there's not going to be much change. But when you go into kind of fluorescence spectroscopy, so apologies for the lot of data here, but this thing here is known as an emission spectra or a fluorescence spectra. Note that it's very similar to a sort of absorption spectra, except for the fact that the fluorescence spectra occurs at slightly higher wavelengths compared to absorption for any molecule, the same molecule actually. So this is tryptophan emitting at around 350 nanometers of wavelength. And what I was trying to do is that in this work was that we were trying to add this hemine molecule and trying to see its interaction. Because fluorescence is more susceptible to interaction, you see that as I keep on increasing the concentration of hemine, the sort of intensity of fluorescence goes down. And ultimately, if you compare this with this, you see there is a distinct shift in energy. And this shift in energy, as you know, can be used to find out the sort of interaction energetics between the system. Now, remember I told you about time resolved fluorescence. So you can actually study the decay of this fluorescence as a function of time. It looks something like this. This is intensity plotted in log scale, and this is time. And this kind of instrument that you use to measure this is a time resolved spectrophotometer, fluorimeter. And when you excite the system at 280 and study the emission at 350 as a function of time, you end up with profiles like this. Note that this is not exactly similar to the kind of textbook level of exponential decay that you see. This is because of molecular processes sort of complicating this, but you can fit it to multiple exponential systems. And then using this, you can find out, let's, for example, the time scales of interaction between molecules. This is a practical example where I found that fluorescence spectroscopy gives you more information about interactions compared to UV visible spectroscopy. And following this, I come to another part, which is standard, again, a sort of absorption kind of experiments, which is vibrational spectroscopy. So again, for those of you who have studied about FTIR, you know about the source and detector, you know about the geometry of data collection, which is again, mostly transmission, and you know about the physical model behind vibration. Physical model is the simple harmonic oscillator. Recall your quantum chemistry course, apply an harmonicity correction to it, solve the Schrodinger equation to find out the vibration energy levels, and then use this to find out vibrational selection rules. This is the sort of basis of vibrational spectroscopy. However, there's a sort of small catch to this. So all of you have heard about what is known as Fourier transform for infrared spectroscopy. So we don't say Fourier transform in the case of UV because the instrumentation is different. So what happens in the case of a Fourier transform machine? So this is a sort of schematic of an IR spectrometer, a modern IR spectrometer that you might be having in your labs. So there's an infrared source, and there are two mirrors here. So one is a moving mirror, and the other one is a stationary mirror. So there's a beam splitter which divides the beam between these two sources. And because of the interaction between the light reflected from the moving and the stationary mirror, what arises is known as an interferogram. So this is known as a Michelson interferometer for those of you who are aware. And this interferogram essentially is then sent to the sample. And the sample interacts with basically this sort of packet of radiation, and then it goes to the detector. The advantage of using this is that you get information about all wavelengths at the same time. And then once you have the interferogram, you apply a Fourier transform, which is a mathematical kind of technique, which helps you transform from the time to the frequency domain. And you use this to find out spectra. And the advantage of using a Fourier transform in IR is that it helps you acquire data much quickly, and it gives you much better spectroscopic resolution as opposed to a UV instrument where you actually scan wavelengths and collect data for each wavelength separately. The question that you may want to ask is that why are Fourier transforms not used in UV's visible spectrometers? That's something that I'm not very clear about, but why don't you give this a thought and look it up in the literature if you can find something about it. A small sort of addition to this is that, modern IR instruments almost always use ATR. ATR is fairly, simple, where basically you have a crystal, you put a drop of your sample on this and on the crystal, and then you sort of, what happens is that there's an ATR crystal and there's the IR beam that goes into the crystal, and there is some sort of total internal reflection occurring within the crystal. This part of the physics is essentially optics, and this is complicated. This is fairly complicated. But suffices to know that when the radiation gets reflected within the crystal, there is a phenomenon which is known as evanescent waves, which are generated on top of the crystal. These evanescent waves can then sample which presumably are kept on top of the crystal, and using this you can find information about bond making, bond breaking, so on and so forth. Summing up the IR side of things, it is a technique based on Fourier transforms, a very important tool in spectroscopy, which you will use later in some other techniques as well. It is used to probe bonds and vibrations, and also bond making and bond breaking. Keep in mind that vibrational energy levels are closely spaced compared to electronic energy levels, so they require IR energy, which is lower in energy compared to UV visible, which is higher in energy. And ATR is the sort of modern tool of choice for doing IR spectrometry in most cases all over the world. Now, following this, I'll come to a quick case study. So this is a report very recent, came out last month in the Journal of the American Chemical Society. And this was a report by 3 groups, one from the University of Yale, from Yale University at New Haven, Connecticut. Another was from the University of Basel at Switzerland, and the other one was at British Columbia in Canada, in Vancouver, Canada. So there were 3 authors in the paper. So what they did was that there was the silicon crystal. This is again essentially IR spectroscopy. And there's a silicon ATR crystal on which they've deposited gold. And on top of that, they've attached these kind of molecules, which are known as, you might be aware of these molecules. This is 4-mercapto benzoic acid, which has been deuterated. And this is 4-mercapto benzonitrile. So these molecules were grown on a gold film. There are ways of doing this. Chemists do this, people who do work in surface chemistry. And this was then put on the ATR crystal. And they studied this with the help of IR spectroscopy. Now, what is this other thing that I've shown? I'm sorry for this detail. You don't need to worry too much about it, but these are electrodes dipped into the system. I'll be coming to this a little later. So there are 3 electrodes, 1, 2, 3. As you probably are aware, with a little bit of introduction to electrochemistry that you have, electrodes are used to apply potentials to the solution. And these potentials can make some changes to this. So this, as you see, is a real life IR spectra. And you plot it as absorbance versus wave number. Now, just a quick word of a caveat, if you will. Usually you're used to seeing IR spectra as transmission versus wave numbers, but in most cases in the literature, if you're interested in doing some kind of quantitative analysis, then plotting it as absorbance makes things much easier. The transformation between transmittance and absorbance is sort of very trivial, and I'm sure you'll be able to look it up in any standard textbook. So this is the sort of IR spectra that these authors were able to collect at this paper, at this report. So you see, this is the sort of stretching frequency for the CN. So again, this is a probe that the CN functionality is present. These are CO stretching frequencies for the COOD, the ones that are marked in purple. Note that the protonated carboxylic acid appears at a different frequency from the deprotonated carboxylic acid. So again, there's a proof that IR spectroscopy is essentially used for studying functional groups as well as subtleties associated with functional groups such as deprotonation and protonation. And these are vibrational modes associated with the benzene ring. A real life example of IR spectroscopy used to solve a sort of research grade problem. There's something interesting, something more interesting that they did was that, again, apologies if this seems a little too complicated. So what they've done is that they've applied potential to the system and they've studied the current. This thing is known as a voltammogram. It's a little bit sort of an esoteric thing associated with electrochemists. I'm an electrochemist, so I'll be trying to push this in one of the slides. But yeah, so effectively what you do is you apply a potential and you look at the current in the system. Looking at the variation in current allows you to follow chemical changes that have been induced because of the potential that you've applied. So at these kind of positive potentials, gold gets oxidized, and these kind of negative potentials, the SAM, this is actually known as self-assembled monolayer, sort of hot topic if not topic in the literature nowadays. So what they've done is they've taken this IR spectra of the molecule, then they've added a potential to the system. The plot here shows the variation in the spectra as you apply positive potentials, which is more towards the red side, and negative potentials, which is more towards the blue side. Note here, without going into too much details, is that as you apply a positive potential to the system, what you're doing is you're stripping off the H, the sort of proton from the carboxylic acid. So effectively, you're increasing the sort of COO minus concentration in the system. This results in a gradual increase in the absorbance as you go towards positive potentials, and a fall in absorbance. Negative absorbance means essentially absence of this molecule as it goes towards more negative potentials. The opposite thing happens for the COOH signal. When you're adding the proton to the system at low potentials, the absorbance increases. Whereas when you're removing the proton from the system at positive potentials, the absorbance decreases. So you see there is a kind of an inverse relation between these two peaks, because this is for the protonated signal and this is for the deep protonated signal. Now, apologies if this seemed a bit too complicated, but the reason why I showed this was that this is one of the examples of in operando spectroscopy. So for those of you who will be practicing spectroscopy, in operando is sort of one of the core areas of research where basically you study a system in operation. So this is different from studying a system in equilibrium. So for example, if you're doing work on solar cells, if you're doing work on batteries, if you're doing work on let's say something else, which is a device, then you need to understand how chemical structures change as the device is sort of functioning, right? So that kind of an experiment when you study spectroscopic properties in tandem with the performance of the device are known as in operando experiments, and these are crucial when you're doing your sort of field reports or class reports on spectroscopy. It would be worthwhile reading up a bit about in operando experiments and what are some of the sort of common in operando experiments that are done in the literature now. Right, so this brings us to the next stop for the day. I think I've been going a little bit slowly, but I'll be happy to speed up if time is a factor. But till now, again, sorry for not stopping before this. I've talked about absorption, I've talked about emission, and I've talked about these in terms of vibrational spectra, about UV visible absorption, I've talked about fluorescence emission. And I've also talked a little bit, I've sort of discussed two case studies where the first one showed why emission spectroscopy sometimes can give a little bit more information about absorption. And the second one has been a sort of in operando study. So till now, apologies for a lot of this. This is not bite-sized, but do you have any questions that you would want me to address or answer before I move on to the next part of the presentation?
I have a clarification to seek. When you say in operando, I interpreted it, it's like you are running the spectroscopy while you are perturbing the system at the same time. Is my interpretation correct?
Yes, Professor Shamasa, that's correct. It's kind of like perturbing the system, that is allowing the system to operate as it is. For example, you have your cell phone, and when you're using the cell phone, it's getting discharged, right? Parallelly, let's say you're also taking a look at the health of your battery inside to find out what's the change in the structure of your battery material as the cell phone is getting discharged. Doing this together in tandem is in operando. So,
Dr. Shamas, there are a few questions are there in the chat box. So
yes, please.
May I
please,
repeat
Dr.
it
Roy.
for your information?
Please, Dr. Roy. Thank you.
Akshay Kumar Moria, he has asked one question: what is simulated emission and spontaneous emission?
Right. So, hi Akshay, thanks for your question. Apologies, I will not be able to elaborate too much on this for two reasons. The first reason being that this would require a lot of math, and second, in the sort of interest of time. So the difference between simulated and stimulated, sorry, I keep on missing these two words. The difference between stimulated and spontaneous emission is that, for example, there is a ground state and there is an excited state. So you raise an electron, or you raise a particle, let's say, from the ground state to the excited state. And because the particle cannot remain indefinitely in that excited state, it has to come back. So this coming back is stimulated by the fact that you've actually raised it to that excited state, right? So this is essentially stimulated emission. Sometimes there may be decay from excited state spontaneously, and that is known as spontaneous emission. Again, like I said, this is more concerned with lasing. So if you read about the physics of lasers, they're very nice, sort of simple but elaborate articles in Wiki as well as standard open source areas. You'll be able to find out that this process of spontaneous emission is more associated with lasers, and the physics is a tad bit different and a little bit more complicated. But I'll just let it stop at that, because going beyond this is sort of beyond the scope of the lecture. Thanks.
Thank you, Dr. Shamasda. Purva Bandyopadhyay, she has asked, which source is used in spectroscopy?
Thanks. Hi, Purva. Thanks for your question. Sort of source meaning, for example, if you're using a UV visible spectrometer, then you would have a UV visible light. So these are usually deuterium, hydrogen. There's a visible light, and the UV light comes from deuterium or halogen lamps. If you're using an IR spectrometer, then you need an IR source. Now, offhand, I can't recall what a modern IR source is, but I remember from my undergrad days at Presidency College where we were reading about IR from William Kemp's organic spectroscopy. So there I had read about things known as I think it's known as a Nernst glower. So that's sort of like the tube which is used to generate IR. Then, for example, if you're going to look at some other kind of spectroscopy, like let's say X-rays, then you have a source of X-rays, which is a Coolidge tube. So the source varies depending upon the type of spectroscopy that you're doing. Thanks.
Then Arindam Roy is asking how Lambert-Beer law is acting in absorption and emission.
Thanks, Arindam. That's an interesting question. The absorption part of the question is, I guess, simple, because when you have absorption of radiation by a molecule, then the sort of radiation going in I0 and the radiation coming out I is connected by the sort of attenuation physics. I is equal to I naught e to the power minus epsilon Cl. You sort of do a little bit of math on this, and you basically frame Lambert-Beer law, where absorption is equal to epsilon Cl. So essentially, in absorption, the sort of amount of light absorbed is directly proportional to the concentration of the what do you call that, like analyte or absorbent, if you will. And this is essentially Lambert-Beer law. Now, if you go to the other side, that is the amount of light emitted. Now, you can argue that the amount of light emitted is also proportional to, let's say, the concentration of the species which is emitting. So note what I'm trying to say here, if I'm making myself clear, is that the amount of light absorbed obviously depends on the concentration of the absorbing species. This is direct Lambert-Beer law, validated from TDPT2. Now, the amount of light emitted, whether this is proportional to the concentration of the emitting species. Now, there's a caveat here that ideally, yes. So emission is also proportional to the concentration of what in chemistry is known as the fluorophore. That's the thing which gives off the fluorescence. However, Lambert-Beer, mind you, is a linear law, right? So it is valid only under specific kind of conditions where the molecules are not interacting, so on and so forth. So the dependence of emission versus concentration, to be honest, I don't know if it is going to be linear at all points of time. At certain points, definitely. I remember doing some kind of emission spectroscopy in my MS thesis, where I was studying the emission of tryptophan in the presence, the emission of myoglobin in the presence of ethanol water mixtures. And I had seen that by increasing the concentration of myoglobin, the emission intensity, if you sort of integrate the area under the emission peak, it does sort of follow a linear regime initially, but beyond a certain point of time, it becomes non-linear. So I am not, to be honest, very sure if Lambert-Beer can be thought of in terms of emission. But at certain points of time, I guess it can be approximated. Thanks.
Can I make a comment here?
Yes, please.
I'm going to draw an analogy to macroeconomics. You see, there is this concept called GDP, gross domestic product. How is that calculated? There are two approaches. You can calculate all the expenditure, the cost incurred in producing the goods, or the income generated. So there are these two parallel approaches, and in order for the system to be stable, they ought to be the same. So GDP calculation. So similarly, it is my qualitative gut feeling that if the amount absorbed is different from the amount emitted, there is going to be, it is going to deviate from the equilibrium. So there is qualitatively, again, it is my instinct says that they ought to be the same. Whether the linearity would hold or not, that's a separate story. But the two curves of absorption and emission should be more or less parallel.
Right. Thanks, Professor Shamazar. Yes, that makes sense. Essentially, emission and absorption are like sort of reflecting each other. So you would see a sort of Stokes shift because there's a slight loss in energy, but essentially they are qualitatively similar. Linearity issues obviously depend upon interaction between the molecules, and so that's a sort of a different sort of ball game altogether. But yeah, thank you.
Thanks, Dr. Shamazar and Professor Shamazar also. Paushali Ghosh, she has asked, what is COOH signal?
Sorry, could you please repeat that, Dr. Roy? Sorry.
What is COOH signal?
So thanks, Paushali, for this question. What is COOH signal? It's, for example, okay, I don't have a COOH model right now with me, but think of it in this way that, you know, there are bonds. For example, this is CO2, right? And you can think of a COOH as basically having a carbon with an oxygen double bond and a single bond OH. So these are bonds, right? They vibrate. So COOH signal is the sort of C double bond O stretching frequency in IR spectroscopy when this is present in the form of a COOH functional group. So I think what I'm trying to say here is each functional group, if you're aware, I'm sure you're aware of the term functional groups in organic chemistry, each functional group with specific bonds have unique IR vibration frequencies, and that's how IR can be used to identify functional groups. So yes, COOH is the vibrational frequency associated with the C double bond O bond when it is present in a carboxylic acid functional group. Thank you.
I make a rather loose common sense analogy. It seems that it is the other number of COOH in the IR world. That's a unique number.
Almost. that's an interesting way of putting it, but yeah, in fact, there is this common term called fingerprinting in IR. So yeah, well, that's essentially your identity in the world of IR spectroscopy.
Thank you. So I think all of the questions have been answered till now. So Dr. Shamasda, right, you can carry on.
Thank you so much. Thanks, Dr. Roy. Just as a sort of, yeah, I just noted that I've almost sort of shooting towards the time limit. I hope this is okay with all of you. I'm happy to speed up a little bit if that's required, but
yes. No problem.
I think I've got a couple of slides more.
You might want to pace up a little bit, okay?
Sure. All right. So this brings me to the sort of, the end of vibrational spectra. Now, quickly, two more things. I've got a problem here. So, sorry, I think this might take a bit of time, but what I'll do is I'll leave this problem to you, and maybe you can just come back to me tomorrow if you have a solution to this. So this is a problem that I've been facing recently, that I faced recently in lab. I was trying to calculate IR spectra, and I had taken this kind of a setup where I had got an ATR crystal and I had a sort of thin layer of gold on it, and I had deposited some material on this and I was trying to look at the IR. However, when I looked at the IR, I was getting only this kind of an observance, the kind of a cosine sine wave, whatever way you'd want to call it. But this is what I got in my spectra. I tried repeating this a couple of times, but this just kept on appearing. Just reminding you that I did have the molecule on top, and this is a very simple regular IR experiment with the gold deposited on a very thin layer of gold, 10 nanometers deposited on the IR crystal. So can you think about what had gone wrong, which is why this kind of a spectra was being obtained? Think about it. I won't discuss the solution right now in the interest of time. Maybe tomorrow I can come back and give you a quick, the next slide has the solution as I quickly sort of skip through it. Just a hint that you know that interferograms after being subjected to a Fourier transform gives rise to spectra. So think about what kind of a signal would give rise to this kind of a Fourier transform. Do a back Fourier, think about a back Fourier and sort of reverse Fourier, if you will, and then try to think along those lines, right? That's a sort of simple problem for you. And quickly, I go to the next one. Sorry for skipping through the slides. So this is about silent vibration. You know, certain things in the world which are silent, but you still need to study it, right? So, you know, vibrations, certain kinds of vibrations are IR silent. This is because when a molecule is IR active, it needs to undergo a change in the dipole moment. There are certain vibrations which do not undergo a change in dipole moment. So these are IR inactive. For this, if we want to study this, we need to use what is known as Raman spectroscopy. So here I'm introducing a new term, Raman scattering. So mind you, sorry, is that scattering is very different from absorption, emission that we've discussed so far. So scattering basically is a photon, is a light photon going and hitting an atom and then moving on a different path. Depending upon whether it's elastic or inelastic, there may be change in the kinetic energy and momentum. So Raman scattering, as you see, is essentially a kind of virtual scattering. So sorry, is a kind of inelastic scattering. So what happens is that here is a sort of electron, and when a laser light goes and hits, it goes to a virtual energy state. And when it goes to a virtual energy state, it can come back to the same state. This is known as Rayleigh scattering. And then what you may have is sort of inelastic scattering, where there may be, it comes back to a slightly higher energy state, or it comes back to a slightly lower energy state. These are known as Stokes or anti-Stokes Raman scattering. So inelastic scattering means part of the kinetic energy of the photon is lost. It involves a virtual energy state, and the signals are very, very, very weak compared to IR. It's 0.01%. So I think the sort of common figure is 1 in around 10 million. But this is important because, you know, using Raman scattering, you can probe bonds and functional groups and bond making, bond breaking for those functionalities where IR spectra, IR spectra is not sort of active. So this is used for those probing those modes which are IR inactive. A quick case study on this. So I was studying, I was trying to look at lithium peroxide formation in this kind of an experiment. So this is the device that I was using. There was lithium and there was a little bit of oxygen here, and oxygen was coming in and lithium was combining, and I was doing a kind of, this is a battery experiment. And again, this is a current potential kind of experiment where lithium oxygen was being formed here, and here peroxide was being formed and lithium peroxide was being broken. Now keep in mind that peroxide has an oxygen-oxygen linkage. So OO dipole stretches, it's a symmetric stretch. So there's very low change in dipole. Because it's very low change in dipole, you cannot study lithium peroxide or lithium or superoxide in IR spectroscopy. So here, this is data that I had collected a year or so back on this thing. This is Raman data. So the OO stretch, which is weak in IR because of low dipole change, is much easier to observe through Raman spectroscopy. So this is a utility of Raman, where, for example, if you're studying a certain kind of system which is not visible in IR, go for Raman. It gives much weaker signals, so you might have to do a much more elaborate or long measurements to increase statistics, but you will get good signals out of it. So this is the progress that we've had so far in my talk since now. Apologies, I've been talking continuously for quite some time, but I'm hoping that you've been able to follow most of it, if not all. So these are atoms, and then there are bonds, and then there are molecules. So I have tried to discuss spectroscopic techniques which can be used to study bonds. For example, IR, Raman spectroscopy. And then there are methods which can be used to study molecules, for example, UV visible and fluorescence. And time resolved fluorescence is sort of kind of an overlapping regime between these two. You can actually sort of like intermediate bonds forming, which results in a change in these molecules, which results in a change in fluorescence. If you study the time, you can, if you do a kind of time resolved study, you can actually, probe the processes overlapping these three. And remember, we had started with a kind of a long, ambitious chart that these are the things that we want to probe. So what we are still lacking is how to probe atoms here and how to probe something beyond this. So when you go from molecules, you can go to these extended kind of crystal structures, but there's something sort of nearby, which is known as kind of a local ordering. So how do you probe this? Quickly, I'll just talk to you about X-rays. So X-rays, as you know, is a sort of high energy kind of radiation. And this is what is known as X-ray absorption. What happens is that X-rays, you can put X-rays, again, this is absorption spectroscopy. You put X-rays in your sample. X-rays can probe much deeper than X-rays. So it can actually go and knock off the core electrons, if you will, from your system. When the core electron is knocked off and it sort of jumps up, what is generated in physics is known as a photoelectron. This photoelectron starts radiating from the atom and it goes and hits the neighboring atom. And because of, apologies for making this sound very simple. It's not exactly this simple, but this is a nice phenomenological way of understanding. The X-rays go and hits the atom. the atom starts radiating photoelectrons. The photoelectrons go and hit the second atom, and this starts radiating something back. And because of this kind of interference, again, note the word interference. This is something that you'll require to use quite a lot when you're doing your crystallography course. And because of this, there's this kind of a fine structure. So effectively, unlike UV visible, where you plot absorption versus energy, this is also, sorry, not unlike, like UV visible, here also you plot absorption versus energy. So this is basically your absorption of X-rays, and these kind of fine structures arise because of these kind of, interactions. So this in total is known as X-ray absorption spectroscopy. This part is known as the edge, and this is known as X-ray absorption near its structure. This part is known as X-ray absorption fine structure, XAFS. And this can be used to probe local ordering along metal sites. The basic principles that are connected with this, when you put in X-rays and the photoelectron comes out, there are other kinds of techniques which you'll probably be reading as a part of your self-learning exercise, such as X-ray photoelectron spectroscopy, X-ray fluorescence, and these are all associated with the basic physics that's sort of represented here. And this is excellent for studying short-range order. So this is the molecule. Now going a little beyond molecule. So this is the short-range order. So this is where this thing comes. However, a caveat that this requires access to what are known as synchrotrons. So quick slide on synchrotrons before I almost end for the day. There's another small case study. So synchrotrons are these amazing areas where basically you take electrons and you accelerate. You might have heard about particle accelerators. There is one in Calcutta at the Variable Energy Cyclotron Center at Salt Lake, just next to Shah Institute. And synchrotrons are sort of the next level of stuff from cyclotrons. So these are like these huge rings. This photo is from Daisy in Hamburg, where I had gone to do a couple of experiments in Germany. And this is from the photon factory in Tsukuba in Japan. Both are amazingly wonderful places to do, amazing science. So what happens is that you take electrons and start accelerating them, and then you make it change its direction by using magnets. So there are things from storage rings, bending magnets, undulators. Read about them in Wikipedia. It's just an interesting thing to know about, especially if you're interested in spectroscopy. But the important thing is that this gives rise to very high energy X-rays. And this is, for example, especially for those of you who are studying material science, if you want to probe materials, you need high energy X-rays. For example, the X-ray absorption experiment that I showed, it requires high energy X-rays. And these are places where you do these kind of experiments. Examples include the photon factory at Tsukuba, DESY at Germany, Soleil in France, the Diamond here in the UK. All these are multinational facilities. If you're interested in performing an experiment from your research lab, you'd be happy to know that the government of India funds visits to all of these facilities. You don't need to spend a penny out of your pocket. You need to design your experiment, submit it to these people, and they do a sort of thorough, rigorous evaluation. And if your experiment is selected, you get to travel there, you get to do the experiment, and you get to collect amazing good spectroscopy data for your research project, research paper, and so on. So this is a sort of very interesting, very, very, very useful set of resources that I used extensively in my PhD. But anyone who's interested, I'd really encourage you to explore these sources and put forward proposals to do experiments there. So and I'll finish off with a small example of synchrotron-based work. So this is something that I was doing basically was that I had studied this hemoglobin and heme. You remember I showed you this kind of UV visible absorption spectra earlier. So this is the X-ray absorption spectra. So this part is known as the near-edge structure, and this part is known as the fine structure. And what I was trying to do, as you see here, the hemoglobin and heme have slightly different near-edge structures. And this is because of the local ordering around these sites, right? And when you actually try to oxidize and reduce the system, you find there are these kind of so this is the derivative plot and this is the absorption plot. You find that there are fine changes that come up between reduced and oxidized forms at the near edge. These kind of changes are not easily visible in UV visible spectroscopy. So the thing that I'm trying to push here is that there's an excellent tool for probing local order in solution. It requires access to synchrotron facilities, but this is also very, very, very useful if you're doing sort of operando experiments, if you're interested in fascinating areas of chemistry like catalysis and so on and so forth. And it gives you much more information than UV visible, especially in the local domain of the chemistry. And this is also a very interesting tool that you can use. There are many other techniques associated with X-rays, such as X-ray fluorescence, X-ray photoelectron, X-ray free electron laser. You can just do a little bit of reading about them online. And they are really fascinating bits of modern spectroscopic tools that really shed a lot of light on the hidden world of molecules, atoms, and materials. So this brings me to the next pit stop, next and the final pit stop. And if you have any questions, but before I'd just like to sort of mark where we stand here. So we have studied bonds and we've identified techniques which can be used to study bonds. We studied molecules and we've tried to identify techniques which can be used to study molecules and the sort of overlapping area between bonds and molecules, because as you obviously know, bonds make up molecules, right? Then we've started looking at short range order in these systems. So as you go further beyond, the next question that probably will be something that I'll try to address in a short bits tomorrow is to look at long range order using a little bit of crystallography diffraction and try to probe something at a much smaller area. If you want to look at the atom, if you want to look at the proton, what is it that you want to, what sort of experiment would you like to design? So these are two things that, just wanted to give you just heads up that these are two things that I'll try to talk about tomorrow. And till then, this is the end of today's talk. And again, do you have any questions up to what I've spoken till now? Questions, comments, suggestions? I'm happy to take. Thank you.
Dr. Shamanta, 2 questions are there.
Yeah.
So one is from Devashish Bandyopadhyay. He is asking, can solid absorption be explained in the light of these type of experiments in case of hemoglobin?
Thank you, Professor Bandyopadhyay, for this question. In principle, yes. I mean, this is probably a sort of very deep area of heme protein research. But yes, the Fourier absorption on the active site of heme proteins, they are uniquely connected to the local coordination environment around the protein. So depending upon, let's say, the type of molecule that is bound to the heme, for example, under the oxygenated state, as those of you in the audience who are aware a little bit about heme chemistry, there's oxygen binding in a particular kind of mode. There are different models of this known as Pauling model, Weissman model, so on and so forth. The geometry of binding. Sorry? I'm sorry, I was not. Janish, could you just
Janish Kumar, do you have any question?
I think it's just a sort of
your voice is
background.
breaking. We could not hear you properly. Dr. Shomaka, please.
So yeah, sorry. And yeah, so it just depends on the kind of ligand binding to the active site, and the Fourier band can be used as a marker to follow the kind of ligands that are binding to the active site of these proteins. Thank you.
Thank you. Another one, Shomodeep Maiti, he is asking, could X-ray absorption spectroscopy distinguish different iron oxide species?
Excellent question. Thanks, Shomodeep. Exactly, it would. In fact, now there's this very kind of nice database on X-ray absorption, which is called, I think, UCARS. Let me see if I can look this up. It's from the University of Chicago. And if you have different iron oxides, including things such as mixed oxides, then using X-ray absorption, you'll be able to sort of find out the concentration of iron 2, iron 3, and so on and so forth. So metallurgy is an area where X-ray absorption is used to a great extent. Yes. Thank you.
Then Mukul Banerjee has asked, field emission simulation using PIC method, how to inject particles properly?
This is a sort of tricky question. To be honest, I don't know the answer because this is a little bit more sort of technical in PIC. I know about injecting of how particles are injected when you, for example, do experiments in a synchrotron. So you have an undulator which is sort of directing the particles in your beamline. But to be honest, I'm not aware of how to inject particles in a PIC, but would encourage you to look this up on the internet if you will, and that'd be like nice. Sorry.
Thank you, Dr. Shamanta. All of the questions have been answered. So thank you for your nice, informative presentation.
Thank you.
hopefully all the participants will be benefited with your talk. So again, participants, if you have any query still remaining, then you can ask it to the experts. If any question comes in your mind later on, then you can share it to us and we can share it to our speaker and we can answer it at its convenient time. So please ask if you have anything.
Exactly. If you have any questions, I'm happy to answer them now or later on. If you would be happy to share it with Dr. Roy, and you can send me an email. There's just one thing. I'm happy to share my slides with you all. Maybe tomorrow I'll just sort of take them up in a PDF and share them with you. And I've taken the liberty of getting a handout ready, just to sort of as a sort of
just about
kind of like a general. Sorry, I think. Yeah, so I'll just get a sort of handout ready on this and I'll just give it to you. It contains a sort of small set of references and sort of guidelines on a few topics that you can study as a part of your self-learning program. And I'm happy to share this with you tomorrow, as well as a set of self-practice exercises. So those are 2 or 3 things that are coming your way from me. But yeah, apart from this, any questions, happy to answer them, email, online, whatever. And for tomorrow, I hopefully will not take as long a time as today. I'm sorry for really eating into your weekend, Saturday lunch time almost. But yeah, tomorrow I'll be talking a little bit about crystallography. I'm not an expert crystallographer, so it'll just be a very top level view. I'll be talking a little bit about NMR and a little bit about electron microscopy maybe, and a few associated techniques. And I'll be sort of happy to wind up with sort of final map for what to study, depending on what you want to see. So yeah, thank you very much for listening to me. I'm happy to have spoken with you, and
I'll interrupt for a second. Okay, here, both Roy and Samajda, let us hang in for a minute or so. So Vice Chancellor called me and he says he's going to join in just for a week. Okay.
Sure. Happy to. Yeah. Right.
We are getting some comments in the chat box, so that's good to know. There is somebody, Jyoti Chaudhary, says that really enjoyed the session. Debashish Bandyopadhyay said it was a lucid explanation. And now we are honored to have our Honorable Vice Chancellor, Professor Saikat Maitro. And this has been a session on the series, the last part of the series on materials characterization. And the first part of this talk concludes today. The talk has been delivered by Dr. Rudra Samajdar, who is a scientist at the National Physical Laboratory, United Kingdom. So that's he delivered his talk on light matter interaction, basically on absorption, emission, diffraction, and phenomena like that. So Professor Maitro, if you would say a word or two.
Thank you, Dr. Samajdar. Actually, I would have been delighted to join at the beginning of this program as a customary measure in other programs as well, but I had to join in 2 other programs simultaneously. However, I believe that this was a wonderful session, particularly from the deliberation from the experts, all the participants. They have enriched their knowledge. Today's topic was on optics powers, this light diffraction, absorptions, and all these things. But this is very important in the context of this emergence of photonics and Li-Fi and all these concepts where optical phenomena will be one of the predominating affairs in our technological this arena. So we need to get a mastery over this optics and addition to this electronics, because this world is moving towards in that direction. This strength, this strength and opportunity the photon is having for this transforming the entire technological scenario, be it communications and in other formats as well, in sensor applications, for changing the face of the world further. So I believe that all the participants they received useful knowledge, and they have this particular webinar is required for sensitizations of the curiosity, hidden curiosity, or latent curiosity in all the learners. And if it has served the purpose, we would be considering that our effort is successful. And taking cue from these webinars, if someone gets interest to further pursue this research and academic activities in these related areas, with the networking and collaborations of all the experts who are present over here, then the purpose of these webinars will be successful. And I believe that in that direction, Dr. Samazdar and all the organizers of these webinars, they have worked painstakingly, effectively to make it happen. So with this, I would like to conclude, because that it was a long session, and now the time is such that everyone of us, they are exposed, all of us are exposed to these numbers of activities in digital world, be it either induced or be it voluntarily taken. So that's why our this attention span is limited. Our ability to multitasking has improved, but whether that has improved the efficiency of doing many things, that is questionable or debatable, but this is the reality. So without this extending further my deliberation, I'd like to conclude with the sincerest thanks to all the panelists who shared their knowledge for the benefits of the learning community, and my best wishes to the learning community for attending these programs and for getting at least some level of enrichment in their existing level of knowledge. And my thanks to Dr. Samazdar and all other faculty members of the Department of Material Science and in the larger context, the School of Natural and Applied Science, for organizing this webinar. So with this, let me conclude finally. I am requesting Dr. Samazdar to say the final words before ending this session. Thank you.
Thank you. Well, you put it so nicely, the real purpose of such events, that if it excites people's curiosity, hidden curiosity and latent curiosity. So yes, that is indeed the purpose. So we have this series, and we appreciate the speaker because the session started at his time. It was 6 o'clock in the morning. So he kept in mind the convenience of the audience ahead of his own convenience. We appreciate the rigor, the references that are shared, and particularly the integrated view that has been presented in this light matter interaction. Would have the second session of this, the part 2 of this series session tomorrow at the same time, that is 11:30 in the morning, where the concluding part, and that I think he's going to focus on the diffraction and few other issues. So thank you very much. With the of Honorable Vice Chancellor, I formally declare this session closed.
Thank you, Dr. Samazdar.
Thank you, sir.
Then I'm leaving. Thank you.
Yes, please. Okay, sir.
So participants, please meet at 11:30. So thank you.
Thank you
Thank you, Dr. Samazdar, also.
very much, Dr. Roy. Thank you. See you tomorrow.